everyone, welcome back to Nail the NEET Revision Series. So today it's going to be the 10th day. So we are going to talk about respiration in plants part 2. So before going in for the session, let me know whether you can hear me and see me, and then we'll go in for the session. Let's just wait for 2 to 3 minutes and then we'll start the session. So before that, I would like to tell you something. If you want the notes of this classes, the previous part 1 and part 2, you can get these notes which will be given in the description box. You can download that notes which will be very helpful for you. Stay on until the end of the class. You will get the materials. And those who have not present for the previous classes, part 1 respiration in plants, you can actually look in for the previous video, which is part 1, and then you can come and check in for part 2. So I just want some of you to just reply me whether you can hear me or not. If not, let me know. Yes. And if you can hear me and see me, just reply me with yes. Okay. So I think all of you can hear me and see me. So let's start the session for today. It's going to be respiration in plants part 2. So previous class we were talking about what is glycolysis, very important process. Then I was talking about pyruvic acid decarboxylation. Then I was talking about Krebs cycle, or we can call it citric acid cycle. Today we are going to deal with very important topic, which is going to be electron transport chain. So before going, let me just give you an overview of what we saw in the previous class, and then I'm going to take you to ETC. Okay. So in the last class, we were talking about glucose. I was talking about glucose, and then I was telling that how is this glucose is going to be converted into ATP. And I told you respiration is going to be an oxidation process, very important. And it's going to be a catabolic process, and it's also going to release energy. Yes, so it's going to be exergonic reactions. So in the previous class we talked about all this thing, just a 2 minutes revision of what we seen yesterday last class, and then we'll go on to the next one. Yes, so I told you we talked about aerobic and anaerobic. We are done with anaerobic completely, so I'm going to talk about only aerobic. So first we studied about glycolysis. And glycolysis, can someone tell me where is the location of glycolysis will take place in the cell? We are talking about cellular respiration. It actually takes place in, anybody? Can you just tell me? Last class, those who were present, you can just answer me. It is present in the cytoplasm. Okay. And then what exactly happens over here? One molecule of glucose is getting converted into 2 molecules of pyruvic acid. And then the second one we have seen, it's going to be pyruvic acid decarboxylation. Now, where is this one has taken place? This has taken place in mitochondrial matrix. Matrix of mitochondria. That's why we used to call mitochondria as the powerhouse of the cell. Yes, very important. So here what exactly happened? The pyruvate, which is a 3-carbon molecule, has converted into an acetyl-CoA, which is a 2-carbon molecule. So there are 2 pyruvic acids, so 2 acetyl-CoA with the liberation of 1 carbon dioxide for every pyruvic acid. And the next one we studied about Krebs cycle. We studied Krebs cycle or tricarboxylic acid cycle. We call it TCA. And this also takes place in mitochondrial matrix. Here we studied, to understand it very specifically, there are 2 cycles because 2 acetyl-CoA are present. So 2 cycle will take place. So if you're going to break a carbon moiety, it is going to give you 2 carbon dioxide. So in this reaction we studied, there are 2 carbon dioxide, 1 cycle if you're going to talk about, and 3 NADH and 1 FADH, and directly 1 substrate level photophosphorylation. Very important point, you should remember. Succinyl CoA to succinate, you have to remember that is 1 ATP directly we got it. This is for one cycle. We have calculated it for two cycle. Now, I already told you if ATP is the energy currency, but in glycolysis, we saw there are two NADH formed. So which cannot be used as such, which is an indirect source of energy, right? So what we have to do is we have to send all this NADH into one pathway to make all this indirect source of energy into ATP, or I can say this is going to be like a check. Okay, where you're going to go to a bank and draw the money. In pyruvic acid decarboxylation also, for one pyruvic acid, one NADH is produced. For two pyruvic acid, two NADH is produced. So what we're going to do, and here for one cycle, and here 6 NADH for two cycle, and 2 FADH for two cycle. So what I have to do, 2 NADH from here, 2 NADH from pyruvic acid, and 6 NADH and 2 FADH has to be sent to the inner membrane of the mitochondria in order to produce all of them into ATP. But I'm going to talk about only this mitochondrial things. I'm not going to talk about this NADH which is actually present in glycolysis. So today's class, we are going to deal with ETC. Very easy if you're going to understand ETC and Oxford's together. Just give me a moment, then we'll start ETC. Can anybody tell me the other name of ETC? Anybody? Can you just guess what's the other name for ETC? It's called as ETS also, electron transport chain. Another important thing, it is called as respiratory chain. Respiratory chain. Why it is called respiratory chain? That's the very important question. So I told you we are talking about only aerobic respiration. Aerobic respiration means utilization of oxygen. But whether it is in glycolysis, I told you glycolysis can take place even when there's oxygen present or absence of oxygen, which is a common pathway for aerobic and anaerobic. But in case of mitochondria, usually requires oxygens. So where is the oxygen most predominantly used? 70% of the oxygen are used only in this respiratory chain, or we can call it ETC or ETS. Okay. This is very important. Another important thing you have to see is what does this respiratory transport system has? Okay. If we have to talk about this respiratory chain, there are very important process. I'm going to talk about 5 complexes. And where is this ETC takes place? It actually takes place in the inner mitochondrial membrane. Inner mitochondrial membrane. This is very important. You will definitely get in your examination. So make it a star point in your book or in your note. Okay. I told you this is going to be a cell. Suppose let's consider this is going to be a cell. So this is going to be the cytoplasm. In cytoplasm, what has happened? Glucose has converted into 2 molecules of pyruvic acid, 2 molecules of pyruvic acid. And what exactly happened? Let's draw a mitochondria here. So here what is going to happen? This acetyl-CoA will enter into the cycle, or we can say mitochondria, and get converted into acetyl-CoA and acetyl-CoA. And what will happen? One cycle will take place, another cycle will actually take place. And after that, what's going to happen? So this is the inner mitochondrial membrane. Suppose if I have to draw, this is the inner mitochondrial membrane. Let's draw the mitochondria. This is the outer membrane. And this is going to be the inner membrane. This is the inner membrane. And this is going to be the matrix. So what's going to happen is ETC actually takes place in this region, which is the inner membrane. So please remember, ETC takes place in the inner membrane of the mitochondria, whereas Krebs cycle and pyruvic acid decarboxylation takes place in the matrix, whereas ETC takes place in the mitochondrial membranes. Okay, so let's talk about what is the mitochondrial process that takes place in the inner membrane of mitochondria. I want all of you to listen carefully. If you have any doubts at any point of time when you're studying ETC, please let me know. I'm going to clear your doubts. Okay. Let's draw a mitochondria. I'm not drawing a cell. I'm drawing a mitochondria. So let's consider this is going to be the outer membrane. And this is going to be the inner membrane. Just understand very carefully. This is going to be the inner membrane. So let me mark it the inner membrane as IM. Okay, this is IM, which is the inner membrane, and this is the outer membrane, which is OM, mitochondrial outer membrane. The space between the inner membrane and the outer membrane is called as intermembrane space. So what I'm going to do, I'm going to write it here. Let me write it here. Inter is something between. So it is between outer and inner membrane. So it is called as intermembrane space. Okay. So we know that This is going to be the matrix. This is the matrix. So where two Krebs cycle has taken place. In the matrix, we already know there are 3 NADH produced for one cycle, 1 FADH produced for one cycle, and 3 NADH produced for another cycle, and 1 FADH produced for another cycle, and pyruvic acid decarboxylation 2 NADH is actually present. So all these molecules are actually produced in the mitochondrial matrix only. So all of them are present, but except glycolysis. Glycolysis is actually taking place in the cytoplasm. It has to come from the cytoplasm to the mitochondria. I'm going to tell you that later. Understand pyruvic acid decarboxylation, NADH, and citric acid NADH and FADH are still present in the mitochondrial matrix. And I told you this is indirect source of energy. And there are going to be 5 complexes. I'm going to talk about all the 5 complexes which are given. These 5 complexes are very simple. They are normal proteins. These proteins are embedded proteins. I'm going to talk in detail. One is going to be NADH dehydrogenase. This is called as complex number 1. This is called as complex number 1. And the second complex is called as succinate dehydrogenase. Succinase dehydrogenase. This is complex number 2. Very important. Please make it a star point. You might be getting this question in your examination. The only enzyme that you see in the citric acid cycle, which is going to convert succinate to fumarate. I told you all the enzymes are actually present in the mitochondrial matrix except succinate dehydrogenase. This succinate dehydrogenase is the only enzyme which is present in the inner membrane of mitochondria. Apart from everything, all the other membranes in Krebs cycle are present in the matrix, except this membrane because they are participating in Krebs cycle also. They're participating in ETC also. Okay. What's the complex number 4? Okay, let's write the complex number 3 and then 4. Complex number 3 is called as cytochrome BC1 complex. Cytochrome BC1 complex. The next one is going to be cytochrome C oxidoreductases or oxidases. C oxidases. This is oxidases. This is complex number 4. So these complexes are normal proteins because mitochondria is going to have a DNA which can produce a protein, and all of them are present in the inner membrane of the mitochondria. So all these 4 complexes are actually going to be what? Involving an ETC cycle. But these 4 complexes 1 to 4 are involved in ETC. But the last complex, which we say complex number 5, complex number 5, this complex is called as ATP synthesis. This complex alone is not present in the inner membrane. It is not involved in ETC. It is involved in OXPHOS pathway, which is oxidative phosphorylation. Phosphorylation, the name itself gives you an idea. Phosphorylation means production of ATP. So only in the oxidative phosphorylation there is production of ATP. We are going to utilize all the NADH, all the FADH, and we are going to produce ATP with the formation of oxidation process. That's why it is called oxidative phosphorylation. So please remember, there are 4 complexes which are involved in ETC. Complex number 1 is NADH dehydrogenase. Second one is succinate dehydrogenase. The third one is going to be cytochrome BC1 complex. And the fourth one is cytochrome C oxidases. And the fifth one, ATP synthesis, but it is not involved in ETC. It is involved in oxidative phosphorylation. I just want all of you to just reply me with the S if you understand. Yes. Okay. What are the electron mobile carriers, or we can say electron carriers? Electron carriers which are actually present. Okay, these are complexes. Inside the complexes, there are some electron carriers which are actually present. What are they? The first one you have to remember, FMN, flavin mononucleotide. Flavin mononucleotide. And the second one is going to be very important. I'm going to tell very important things, which is going to be FES, ferrous sulfur. These are all carriers. These are present inside the complexes. Very important. The next one is going to be cytochromes. Cytochromes. So we can say cytochrome complexes. Usually, cytochrome complexes are made up of heme, iron. Usually, they are made up of heme proteins. So they are iron proteins, we can say. And there are different type of cytochromes which we will be seeing, which is going to be cytochrome B. The next one is going to be cytochrome C1. Another one is cytochrome A and cytochrome A3, and then cytochrome C. This cytochrome A and A3, all the cytochromes are having ferrous. This is going to have ferrous. With that, they are going to have copper. So they have copper also. In our book, they have written they are coupled with two copper proteins or copper molecules which are actually attached to it. Very important point. I want all of you to make it a star point over here. Okay. So these are all present, but one question, very important thing you have to remember is which one is soluble in water, which is not soluble in water, which is lipid soluble. We know the membranes are made up of phospholipids. So whichever molecule is lipid soluble, they can be embedded in the membrane. So this is lipid soluble. So I can say all of the things are lipid soluble. Let me make it this way. Let me make it this way. Okay. So this is going to be lipid soluble. So it will be present inside the complex. Whichever is lipid soluble, they'll be present inside the complex. This is also lipid soluble. It will be present inside the complex. And very specifically, cytochrome. Cytochrome is going to be water soluble. So this is like a moving one, which will not be embedded. It keeps on moving. That's why in our book, they have given a straight line where the cytochrome is put in the between of these things. Okay, this is water soluble, so they are mobile. They keeps moving. Another important one, which is going to be ubiquinons. Ubiquinon, or we can call it CoQ. This is also going to be water soluble. So if they are water soluble, they are also going to be mobile. So there are two of these things, mobile carriers, electron carriers, which are mobile in nature. One is going to be ubiquinon, another one is going to be cytochrome. Okay, yes. So let's write all the things together. So let's talk about what is oxidation and reduction. Without understanding this concept, you cannot understand ETC. So please make sure to understand what is oxidation. And let's talk about what is reduction. And then directly we'll go on to the calculation part. Okay, listen. So oxidation, please remember oil rig. Oil rig. So oxidation means loss of electron. Very simple. So loss of electron. Reduction means gain of electrons. So it's going to be gain of electron. This is electronic terminologies if you're going to say. Or else we can say addition of oxygen, or we can say removal of hydrogen. Removal of hydrogen. And reduction means addition of hydrogen and removal of oxygen. So here we are going to use two important things which is very important for ETC. One is based on electronics. So oxidation is loss of electron. When a molecule is going to lose its electron, which means it is getting oxidized. When a molecule is going to get electron from somebody else, then it is getting reduced. Another important thing you should remember, removal of hydrogen and addition of hydrogen. These two are very important in this case, and these two are very important in this case. And always listen, electron always used to keep on traveling. But how will an electron travels? Sometimes in a question paper, they can give you reducing equivalence or reducing power. So you just remember reducing equivalents are very simple, which are nothing but they are electrons. They are simply electrons. Okay. So when will the electrons actually move? Suppose let's consider there is one molecule, or let's consider one person in a class. So if a person in a class is very, very active, or we can say he's having high energy, that person will not sit in one place, right? He used to be keep on wandering because he's very energetic. So any molecule which has high energy are going to be unstable molecules. So make sure electrons travels from high energy to low energy. Important concept, applicable for photosynthesis also for respiration. High energy to low energy. Why an electron has to go from high energy? If anybody is going to have, or we can say a molecule is going to have high energy, if anybody is high energy having it, they will be very unstable. Nobody wants to be unstable, or we can say no molecule want to be unstable. So what they try to do, I don't want high energy, I'm going to give some of the energy to somebody else, so they become stable molecules. So electron always travel from high energy to low energy. Another important thing, electron travels from low redox potential. Very important, low redox potential. We already know what is redox, which means If oxidation and reduction takes place simultaneously together, we used to call it redox. A molecule which has a redox potential are going to be excellent donors, electron donors. So please remember, suppose if electron is transferring from complex 1 to complex 3, which means complex 1 is going to have low redox potential. That's why they are giving electrons to complex number 3, because they are excellent donors. So please remember, electron travels one thing from high energy to low energy, or from low redox potential to high redox potential. Important concept for your electron transport chain, not only in respiration, also for your photosynthesis. Please make sure to understand this concept very clearly. Okay. Now we will understand this one in detail. Okay. Yes. So let me write it over here. So I'm going to remove certain things. Okay. For our understanding, we already know there are 4 complexes in ETC and 1 complex in oxidative phosphorylation. So I'm going to talk about all the complexes over here. So let me make it, this is the inner membrane. So let me write it as this is complex number 1. And I'm going to write complex number 3. I'm not writing complex number 2, and I'm writing complex number 4. And I'm writing complex number 5, but this is not involved in ETC. It is an oxidative phosphorylation. For now, we are going to deal only with 4 complexes. For our understanding, I'm writing the complex number 2, which doesn't mean it is actually present at the bottom. It is actually nearby. For our convenience, I'm writing it here. Okay. We know that complex number 1 is called as NADH dehydrogenase. So what can happen is whatever the NADH which has been produced in Krebs cycle, or we can say pyruvic acid decarboxylation, will come over here. So NADH plus H plus. Okay. So now what is going to happen? They are going to lose electron. Okay, loss of electron is called as oxidation or reduction. Yes, hi Mohammed Rizwan. Oxidation is called as loss of electrons, a gain of electron, loss of electrons. And we know reduction, suppose if I have to talk about reduction, gain of electrons, or we can say addition of hydrogen. And oxidation, just now we have seen, it is going to be loss of electrons and removal. So what I'm going to do is, I'm going to remove NADH2 from there. I'm going to remove 2 protons from there, or 2 hydrogens from there, which means NADH is getting oxidized. Okay, so what I'm going to do, from NADH plus H plus, I'm going to remove 2 protons out. So I'm removing a hydrogen, which means it's going to be oxidation process. So NADH is getting oxidized. Very important. In your book, they have given oxidation of 1 NADH is going to give you 3 molecules of ATP. I'm going to deal in detail. So remember, NADH is getting converted into NAD plus 2 protons. Okay, there has to be loss of electron because oxidation. Already we have removed this hydrogen and the electrons are lost. So who is going to accept this electron? So electrons will always move from high energy to low energy. So NADH is going to have high energy, complex 1 is going to have less energy. So they move from high energy to low energy. Or we can say NADH are excellent electron donors, which means they are at low redox potential. So what exactly happened? The electron is given to complex number 1. And I told you inside a complex, there are some proteins present, which are going to be FMN and FES. These are some of the proteins which are present inside it, and I told you they are going to be lipid soluble molecule. That's why they are embedded here. Now what will happen? The electrons will be accepted by FMN. Okay, accepting an electron, it's not gain of electron. Now what will happen? FMN will change into FMNH2 while accepting electron. Now this is getting what? Reduction. So can I say oxidation and reductions are taking place simultaneously? So this process is called redox reaction. So what is happening? NADH2 is actually giving electrons and removing protons. So this is oxidation. NADH is getting oxidized, whereas the molecule which accepts electrons and adds hydrogen are getting reduced. And now this FMN is going to give electrons, pair of electrons to FES. Now, what's going to happen? I told you there are 2 molecules which are going to be water soluble. One is going to be ubiquinone, another one is going to be cytochrome C. Very important. So I'm going to write ubiquinone, which doesn't mean they will be present here. So ubiquinone keeps on moving in the inner membrane actually. But cytochrome C is present on the outer surface. Just a moment. It's present on the outer surface of, which means they are not present in the intermembrane space. They are nearby the inner membrane. These two molecules are water soluble. That's why they are not present in the membrane. They're actually protruding out of the membrane because they are water soluble, right? Yes. Now what will happen? This electron is given to ubiquinone. When they get electron, what will happen? It becomes UbQH2. Okay. And then this ubiquinone goes to complex number 3. Before going, what will happen? This proton is there, right? This proton from the matrix will be pumped into the intermembrane space. So let me make it here now. The proton has come, 4 protons. Usually 4 protons will come over here, which will come to the intermembrane spaces. 4 protons will come to the intermembrane phases. For calculation, I'm writing it. So first complex, after the electrons are getting transferred, 4 protons are pumped if the ETC cycle starts with NADH. Now the electrons are still present in ubiquinone, which means they have reduced UbQH2. What is the second complex? Second complex is called as succinate dehydrogenase. And I told you, this is the only enzyme which is present in the mitochondrial matrix in the inner membrane, which is involved in both Krebs cycle also, ETC also. And what will happen in succinate is getting converted into fumarate. That time, very important molecule will be produced. What is that? FADH. Now, here what will happen? FADH will come, DH2 will be oxidized. Yes, oxidation is going to take place, FAD plus 2H plus. Now, what will happen? They will give electrons here, but who is going to get the electrons? FES. FES gets the electron. Now, this electron will also go to ubiquinone. So, electrons from the complex 1 will also go to ubiquinone. Electron from complex 2 will also go to ubiquinone. Okay, yes. Still now we have seen if the ETC starts with NADH, 4H plus is produced. But FADH, if it starts, it will not go to complex 1. It will always go to complex 2. So 4H plus will not be produced. Let's do next. Next, the electron will be given to complex number 3. Now, what's going to happen in complex number 3? Complex number 3 is called cytochrome complex. So there are 3 electron carriers, cytochrome B, cytochrome C1, and in between there is going to be FES. So what will happen? The electron will come to cytochrome B and the electron will come to FES and the electron will go to cytochrome C. Okay. After that, this electron will go to cytochrome C. Now what will happen? Now 4 protons will be pumped into the intermembrane space, not in the outer membrane or inner membrane, in the intermembrane space. In ETC, all the protons which are present in the matrix will come into the intermembrane space. Okay, now let's do a calculation. If the ETC cycle starts with NADH, first 4H plus has gone and then second 4H plus has gone. But if the ETC starts with FES, first 4H plus will not come. So it goes only to complex number 3. So only 4H plus will come. Okay. Now what will happen? Cytochrome C is going to give its electron because they are at high energy. They don't want to be at high energy. They want to give it to somebody else. So what will happen? Complex number 4. Complex number 4 is called as cytochrome A, cytochrome A3 complex. And this is a very important small protein actually. It is given in your book, very important, and it is having copper. 2 coppers are actually present and it is going to be a mobile carrier. Okay, what exactly happened? This cytochrome A is going to get electrons and cytochrome A3 is also going to get its electron. Now, who is going to accept the final electrons? That is going to be oxygen. So, oxygen is going to be the final electron acceptor in a ETC cycle. So, it's going to be half 2H plus 2 electrons. So, which is going to produce water molecules. Yes. So, in ETC, there is a production of water molecules. Why? Because they used to have nascent oxygen and some protons are present here. They accept the electrons from cytochrome A3. And now this time, 2H plus molecules will be going into the intermembrane space. So let's do the calculation. I'm not talking about Oxford's pathway. Let me revise it once again. All of you listen. I told you that ETC cycle is actually taking place in inner membrane of mitochondria. There are 5 complexes. Complex number 1 is called NADH dehydrogenase. Complex number 2 is called succinate dehydrogenase. Complex number 3 is going to be cytochrome B1C complexes. 4 is going to be cytochrome C oxidases. I'm not talking about fifth one, which is involved in oxphos. So if NADH produced by pyruvic acid decarboxylation, or if it is produced by the matrix through Krebs cycle, all will be present in the matrix. Now this NADH will not go to complex number 2. They will go to complex number 1 because it is NADH dehydrogenase enzyme. So NADH is getting converted into NAD+, which means they are going to get oxidized and they were going to lose electron because oxidation is loss of electron. This electron will be carried by FMN and then they will get, after accepting electron, they get reduced and they give it to the FES. And after getting these things, four protons will be pumped from the matrix to the intermembrane spaces. And if the ETC cycle is going to start with FADH, FADH will never go to complex number 1. They will always go to complex number 2. So that time, this electron will also go to ubiquinone Q. And then the electrons from complex number 1 and complex number 2 will go to complex number 3. And then they will go to cytochrome C. And then they will go to complex number 4. And the final electron acceptor is going to be oxygen. The question paper, you will be asking who is the final electron acceptor in ETC cycle. Where is the maximum oxygen used? It's going to be respiratory chain, or we can say ETC. Okay, now let's do the calculation of this ETC. Okay, I'm going to count this one, very important for your examination. So question in your book, there is oxidation of NADH2. Why? Because whenever you're going to oxidize NADH2, it becomes NAD plus 2H plus electron, and this electrons only will be given to each of the molecule because high energy to low energy, low redox potential to high redox potential. So whenever this oxidation, it goes to complex number 1, that time 4 protons are pumped into, very important, intermembrane space. From where it is going? From matrix, it is going to intermembrane spaces. It is going to intermembrane space, and then 4 protons after complex number 3, and finally 2 protons are going to finally. So totally there are going to be 10 protons which are pumped from the matrix to the intermembrane space. Very important, intermembrane space, if the ETC cycle starts with NADH. But oxidation of FADH2. Suppose there are FADH2 produced in Krebs cycle. Nowhere else it is produced. 2 FADH are produced for 1 glucose molecule or 2 Krebs cycle, we can say. This is getting converted into FAD+, which means it is getting oxidized. And 2 protons and 2 electrons. These electrons will be given. At that time, the first complex, no protons are going into the intermembrane space. So second, 1 proton goes and 2 protons. So totally, it's going to be 6 protons only. So you should remember, if the ETC cycle starts with NADH, there are 10 protons which are pumped from matrix into the intermembrane space. If the ETC cycle starts with FADH, the protons which are pumped into the intermembrane spaces, all are now in the intermembrane spaces. Please remember, all are now in the intermembrane spaces. I'll tell you why there is a need to go from matrix to intermembrane spaces. Again, when they'll come from intermembrane space to matrix again. That time they'll produce energy. That's the purpose actually. Now they are in the intermembrane spaces. Please understand, there is a very important line on the last page of this Oxford's, the last 2 lines. Every, or we can say, for 4 protons, very important, for 4 protons, 1 ATP will be generated. 1 ATP is generated. So let's calculate. I just want some of you to just answer me whether you can understand or not. Okay, 4 protons. So 1 ATP here, 1 ATP here, and 2 protons. So it's going to be half ATP, 0.5 ATP. So totally 1 plus 2 plus 0.5. So it's going to be 2.5 ATP. So we can call it 3 ATP. That's why we used to say oxidation of 1 NADH is going to give you 3 ATP molecules. And oxidation of FADH, let's do 4 protons is giving 1 ATP and 2 protons is 0.5 ATP. So totally it's going to be 1.5 ATP. So if I have to summarize, NADH, oxidation of NADH, not DPH, is going to provide 3 molecules of ATP, a round of value. You should not mark it as 2.5 in your examination. Just mark 3 ATP, which is given in your NCRT. And FADH, one FADH is going to give you 2 ATP molecules. Okay, but we have not produced any kind of ATP in the cell. So next is going to be oxidative phosphorylation. Yes, so let's talk about now. If I have to draw the structure back again. So now we know, suppose let's write it this way. Just a moment. This is the outer membrane. This is the inner membrane. So in this inner membrane, ETC has taken place. But where is the proton now present? The protons are now in the intermembrane space. Why are we pushing all this proton from matrix? Because we are creating a gradient maximum there. If we are pushing all the protons from matrix and now the proton is maximum here. So this space is called as what? Intermembrane space. But I told you oxidative phosphorylation takes place in the inner membrane only, but very specifically in F0, F1 particle of crystal. Now what's going to happen? I told you there is a last complex which is called as complex number 5. And this complex number 5, I'm going to make it this way. This is complex number 5. So there are two proteins which are actually present. This protein is called F0 and this protein is called F1. This protein is actually present inside the membrane, or we can say outer the membrane. So it is called peripheral protein. Peripheral protein. And this is called as, we can say, integral proteins or internal proteins. Integral protein, which means they are attached to the one. And always remember, F1 is going to act as ATP synthesis. So whenever an enzyme is present, there need to be a substrate. So ATP synthesis, F1 is going to act as ATP synthase. Now what's going to happen is, two protons will come, but for our calculation, we are doing 4 protons are going to be giving 1 ATP. But usually, whenever it is pushed into this one, only 2 protons will go, which is going to be 0.5 ATP. So it will go like 0.5 ATP, 0.5 ATP. But for our understanding, I'm just going to write like this way. So what will happen is 2 protons will come over with energy. So when they're going to come to F0 and F1, what will happen is this F0 is going to behave like an enzyme. So whenever an enzyme is present, still now the matrix is going to have some ADP molecules. So what will happen? This ADP and PI, which is an inorganic phosphate, with the help of this energy will get converted into ATP. So this proton again all will come to the matrix. So what we are doing actually is, previously all the protons are in the matrix only, but we are pushing in the ETC cycle to pump everything into the intermembrane space. And from the intermembrane space, we are bringing back again to the matrix so that ATP is actually produced with energy. So please remember, for NADH, if I have to talk about, they're going to produce 3 ATP. FADH is going to produce 2 ATP. So this is the most important thing for your examination. Now we are going to do the complete calculation for the aerobic respiration. Complete calculation, very important. Let's do it for aerobic respiration. This is going to be the energy sheet which is given in your book. Okay, I told you in glycolysis, 4 ATPs are actually produced. 2 ATPs are used in the beginning. So 4 minus 2 is going to be 2 ATPs. So 2 ATPs. And I told you, for one glyceraldehyde 3-phosphate, 1 NADH, another NADH from dihydroxyacetone phosphate. So 2 NADH. And I told you, 1 NADH is going to give 3 molecules of ATP. So it's going to be 6 ATP. So 6 and 2 makes around 8 ATP. In glycolysis, you're going to get 8 ATP. The next one, decarboxylation of pyruvic acid, there is no ATP. Only NADH is produced. So 2 pyruvic acid, so 2 NADH. So 1 NADH gives 3 ATP. 1 NADH, 3 ATP. So 2 is going to be 6. So totally 6 ATP for pyruvic acid decarboxylation. Krebs cycle. Once we already know directly 1 ATP is formed in one cycle, another ATP is formed in another cycle because there are 2 cycles. And 3 NADH in one cycle, 1 FADH. So it is going to be 3 into 3, which is 9, and 1 into 2, which is going to be 2. So totally for one cycle, we can say 11 of this one and 1 of ATP. So 12 ATP for one cycle and another 12 ATP for the second cycle. So totally it's going to be 24 ATP. So if you're going to calculate 24, 6 and 8, it's going to be 38 ATPs. But sometimes in a question paper, they might give you 36 also. Sometimes they might give you 36, 38 also. But I'm going to tell you why our calculation comes around 36 sometimes. That's very important. All of you listen carefully. I told you that pyruvic acid decarboxylation NADH is present in mitochondria. It is also present in mitochondrial matrix. And Krebs cycle NADH what happened? Krebs cycle NADH is going to have one NADH, another FADH. Now all of them are present in mitochondrial matrix. But I told you glycolysis. Glycolysis is going to have NADH produced, two NADH. But listen carefully, they are in the cytoplasm. They are in the cytoplasm. But mitochondria is present over here. So mitochondrial membrane is actually made up of phospholipids. NADH is a polar molecule. So in order to cross that, they have to spend some energy. That's where two ATPs are actually utilized. So we're going to talk about two important systems, which are going to be two shuttle systems. So one shuttle system is called as malate aspartate shuttle system. Another one is going to be glycerol phosphate shuttle system. Glycerol phosphate shuttle system. So let's understand this one. The first thing is going to be, let me talk about malate aspartate shuttle system. I'm writing it as MA shuttle system. So here what exactly is going to happen is, suppose this glycolysis is taking place in a cell. We know, suppose if I have to make this way, so this is going to be the cytoplasm and this is going to be the mitochondria. This is going to be the mitochondria. But we know that here NADH is produced. That two NADH are actually produced. So if two NADH are produced, they cannot cross this membrane. So what exactly happened? For one NADH, one ATP has to be spent. For another NADH, another ATP has to be spent. That's why what exactly happens. So NADH will not go like that. NADH will get converted into NAD plus 2H plus. And this 2H plus only will go inside. Okay. So this NADH will not go. Only the protons will go inside. The protons will go, combines with NAD plus and forms NADH2. So when NADH has to go from outside to inside, what they need some amount of ATP. So 38 ATPs are actually produced, but 2 ATPs are actually being utilized to pump this NADH from the cytoplasm to the mitochondrial matrix. That's why the calculation comes around 36 ATP. In glycerol shuttle system, if I have to talk about glycerol shuttle system, just understand, if I have to talk about glycerol phosphate shuttle system, malate aspartate shuttle system is very efficient. Glycerol is not that much efficient because here also some of them will use that pathway, some of them will use glycerol phosphate shuttle system. Here what exactly happens is the NADH2 will be converted into NAD plus 2H plus. Only the proton can go inside. Only this proton will go inside. That time we know 2 ATP has to be spent for 2 molecules of NADH. So 2 proton. But here in glycerol phosphate shuttle system, FADH is actually formed. So it's going to be FADH2. And we know that FADH2 can produce only 2 molecules of ATP. So that previous shuttle system is the most efficient shuttle system than the glycerol phosphate shuttle system. So when you're doing a calculation, you'll always end up with 38 molecules of ATP. But we know that in a glycerol, 2 NADH are actually formed. They also have to be converted into ATP, right? So they cannot go inside the mitochondrial membrane. For that, for one NADH, one ATP has to be spent. For another NADH, another ATP has to be spent. So 38 minus 2 ATP is going to be 36 molecules of ATP. So your calculation for aerobic respiration is going to be 36 molecules of ATPs are produced for one molecule of glucose. Just understand how beautiful it is. One glucose is actually giving you For one cell, 36 ATPs. In case of anaerobic, we know it's going to produce only 2 molecules of ATP. Anaerobic, when there is no oxygen, it's going to produce only 2 ATP. So we have done with the complete process of the aerobic and anaerobic. So you can just do this question. The overall goal of glycolysis, Krebs cycle and electron transport chain is the formation of what's the purpose? So we are actually, what's the purpose of converting all the glucose? We want to produce an ATP molecules, but it is going to be a small step. So option can be B or C. Just going for rejection option. Is it large oxidation or small step oxidation? It is always going to be what? A small step oxidation. So answer is going to be option B. Okay. You can try with the next question. The energy releasing metabolic process in which the substrate is oxidized without external electron acceptor, which means they do not go for ETC. Only in anaerobic respiration, the molecule will not go in for ETC because mitochondria always requires oxygen. So without electron acceptor means they are talking there is no ETC. Where it will take place? Anybody? Can you answer me? Good evening. Yes, anybody? Sophia, Roshni, can you just answer me? So what's the answer for this question? It's aerobic or anaerobic. The answer is very simple. No ETC. So which is going to be anaerobic. So I'm giving you, yes, very good, Sophia. The answer is going to be fermentation. Okay. Okay, so we have done with the complete aerobic, anaerobic. Now we are going to amphibolic pathway, which is given in a book. Very easy pathway. Only one tabulation, if you're going to understand, or this image is enough for you to complete this process. What is amphi? We already know about amphibians, which means which lives in both the cases. Amphibolic means there is going to be catabolism also. There is going to be anabolism also. We already know about catabolism means breaking down of large molecules into small molecules. Anabolism means combining all the large molecules and making or we can say large molecules, small to large. So here what will happen is some molecules are broken down into small pieces. Small molecules will combine together to form large pieces. So let's talk. We already know carbohydrates, which is going to be glucose. In glycolysis, we said glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-phosphate, and after that, it's going to split into 3 carbon and a 3 carbon. And glyceraldehyde is going to give you pyruvic acid. And then we know Krebs cycle, acetyl-CoA. And there is going to be Krebs cycle. The question can be like, what is the intermediate of aerobic process? Then you should remember Krebs cycle. What is the intermediate for aerobic and anaerobic? Then you have to answer glycolysis, because glycolysis is a common pathway for aerobic and anaerobic. But if the question is asked based on aerobic, then it's always going to be acetyl-CoA. Okay. I told you previously that we don't always use carbohydrates, or we can say glucoses, as our energy source. Sometimes we eat lipids, sometimes we eat proteins. So those molecule also has to be converted into ATP. How that's the case? We know fats are made up of fatty acids and glycerols. What happened to this fatty acids? This fatty acids will be converted into acetyl-CoA. This is very important. Many used to make a mistake. Make sure fatty acids will never convert into pyruvic acid. Fatty acids will always get converted into acetyl-CoA. Just remember, acetyl-CoA itself gives you an idea. It's related to fats. So fatty acids will change into acetyl-CoA. What about glycerol? Glycerol, just remember, glycerol is a 3-carbon compound. So what will happen? Dihydroxyacetone phosphate. So glycerol will change into dihydroxyacetone phosphate. Fatty acids will change into acetyl-CoA. Okay. Suppose if I'm eating proteins, what exactly happened? Proteins are made up of a lot of amino acids. Amino acids are the building block of proteins. We already know. So proteins, all the peptide bonds will be broken and you're going to get amino acid. What happened to amino acid? Even amino acid has to be converted into energy. You eat proteins. So what will happen is deamination process, which means removal of all the amine groups. They will get converted into pyruvic acid, not acetyl-CoA. They will get converted into pyruvic acid and then into acetyl-CoA, and then finally energies will be produced. Okay, so what you have to remember, so it can be any of the respiratory substrate. It can be proteins or lipids. It can be carbohydrates. If they are going to be fatty acids, they will be converted directly into acetyl-CoA. If it's going to be glycerol, they will be converted into DHAP, which is dihydroxyacetone phosphate. If it's going to be a protein, they'll be converted into pyruvic acid and then into Krebs cycle. This is all about amphibolic pathway. So let's do some question. You can answer this question, anybody? Just answer this question. This metabolite, yes, this is important question which has come in 2013 and 16 also. So this metabolite is common to respiration mediated breakdown even for fats, carbohydrates and proteins. What's the answer? Very simple answer, direct question. Yes, what's the answer? Is it fructose 1,6 or glucose or acetyl-CoA, or it is going to be pyruvic acid? Yes, so let me just answer. Okay, the answer is going to be, just now I told you it's going to be acetyl-CoA. So answer is option C. Okay. Next, the last topic, we have come to the end of this session. Only this topic is left. After that, we'll just directly finish off the class. Okay. Respiration quotient. Yes. So what is respiration quotient? This will come in your examination. Very easy if you're going to do it. During aerobic process, this is going to happen only in aerobic respiration, which means in any part of the cells, that too cellular respirations. During aerobic respiration, what's going to happen? You have to remember this formula, which is going to be the volume of carbon dioxide evolves and the volume of oxygen consumed. How much is the oxygen that has been consumed? Because we know respiration means what? Respiration means we are going to take in oxygen because aerobic process. So you're going to take oxygen and you're going to eliminate what, or liberate carbon dioxide. So that is respiration. So how much amount of the carbon dioxide you're going to evolve out and how much amount of the oxygen that you're going to take in. So let's do some questions. See, this is a very important question. They can ask you what's the RQ value of carbohydrate. Sometimes they'll not give you what's the RQ value of a carbohydrate. They can give you what's the RQ value of a starch. They can give you what's the RQ value of potatoes. They can give you RQ value of roti, whatever it is. They can give anything. So just remember the carbohydrates, what is the RQ value? We already know this is going to be the equation which we have done when we were doing our respiration, aerobic respiration. So see how many molecules of carbon dioxide is actually evolved divided by how many molecules of oxygen is consumed. So what's going to be the answer? Very simple. So volume of carbon dioxide evolves by volume of oxygen that is consumed, which is the RQ value. So how to do 6 carbon dioxide and 6 oxygen? So it's going to be RQ value for a carbohydrate. For any carbohydrate, the answer is going to be only 1. Okay, yes. Very good, Roshni. The next one. When we're talking about fats, okay, what's the RQ value for fats? Very specifically in your examination, they'll ask you tripalmitin, fatty acids, tripalmitin they'll ask you. So just remember this one. Suppose if the question is asked for fats, just remember less than 1. But if they specifically ask you, then go in for tripalmitin, then go in for 0.7. You can see one RQ value for this one is 1 or 2 divided by 145. So answer is going to be 0.7. So if it's tripalmitin, it's 0.7. Okay. Proteins. So I told you for carbohydrates, RQ value is going to be 1. And for proteins, we have done with fats. We have done with fats, which is going to be less than 1. Tripalmitin, very specifically, it's going to be what? 0.7. Okay. If it's going to be proteins, so proteins usually have a value which is less than 1. Usually it's going to be 0.9. You can see it over here. It's going to be 0.9. Okay, I'm going to give you another work and you're going to tell me the answer for this question. I'm going to write one important equation. You're going to tell me whether it's aerobic or anaerobic and we're going to deal with it. Suppose in examination, if they give you something like this, C6H12O6, and they're going to give you 2 molecules of pyruvic acid and then 2 molecules of ethanol plus 2 molecules of carbon dioxide. Now they're asking you to calculate RQ value for this one. How to calculate? So how much is the carbon dioxide that is coming out? 2 carbon dioxide. But are you seeing any oxygen in this reaction? No oxygen. Which means if there is no oxygen, this process is anaerobic. In your examination, they not only ask you aerobic. If it's going to be anaerobic, you will write 2 molecules of carbon dioxide and there is no molecules of oxygen. So what's the answer going to be? Anybody, can you answer me? What's the answer? 2 by 0? It's going to be infinity. So very important question. You have to remember what is the RQ value for anaerobic respiration means it's going to be infinity. Okay, yes. Okay, let's do another one. So let's make a tabulation for this one. So RQ value for carbohydrates. So RQ value of a carbohydrate is always going to be 1. And RQ value is less than 1, or we can say zero point 0.7. In case of fats, tripalmitin. I'm writing tripalmitin. And proteins are going to be 0.9, or you can call it less than 1 also. And next one, anaerobic, we have seen it is going to be infinity. Very important. And suppose if they ask you a question like organic acids, any organic acid, usually if you see any organic acid is going to have greater than 1 RQ value. Okay, this is very important. Okay, yes. So we have done with all the respiratory quotient which will come for your examination. Let's talk about few more questions which will come for your examination. Just answer this question. How many ATP molecules could be generated from one molecule of glucose? Yes, we are talking about one molecule of glucose. One molecule of glucose is going to produce how many ATPs? But it is conditional. We know it's going to be 36, but here we are not referring it there because they're talking about complete oxidation. And I told you glycolysis is partial oxidation, aerobic is complete oxidation, anaerobic is also partial oxidation. Complete oxidation of one molecule of glucose is going to produce carbon dioxide and water. We know carbon dioxide is produced in Krebs cycle and pyruvic acid decarboxylation, and water is actually formed in ETC as well as in glycolysis. Okay, and they are going to give you an energy of how much they are going to give you an energy? It is going to be 686 kilocalories. So one glucose is going to give you energy of 686 kilocalorie. And it is available in the high energy phosphate bonds. 1 mole is going to 1 ATP, 1 mole of ATP is going to give you 12 kilocalorie. And 1 glucose is going to give you almost 686 kilocalorie. Now you have to find out how many ATP molecules. So can anybody answer this question? Anybody? Just answer this question. What would be the answer for this question? Okay, very simple. If you have to calculate, just divide 686 divided by 12, which is going to be 57. So answer is going to be option A. Okay, the next question. During which stage is the complete oxidation of glucose is the greatest number of ATPs produced from ADP? Where ATP is actually produced? Maximum ATP. Can anybody answer me? Where maximum ATP is produced? Very good, Sophia. It's going to be electron transport chain. If you talk about glycolysis, only 2 ATP. Pyruvic acid decarboxylation, only we can say no ATPs are actually produced. If you're going to talk about Krebs cycle, 2 ATPs are produced for 1 glucose. But in electron transport chain, 1 NADH itself can give 3 ATP. So we saw there are so many NADH which is coming to the ETC. So maximum ATPs are produced only in electron transport chain. Suppose if the question comes like Oxford, then you can mark it that way. Okay. The last question, chemiosmotic coupling hypothesis, which you have studied in photosynthesis. Oxidative phosphorylation has proposes ATP is formed. Why the ATP is formed? I told you the protons are actually in the intermembrane spaces and then they come to the matrix again, which means we are creating what? Proton gradient. So what is the answer for this question? Anybody answer? This is 2008 question. Anybody answer this question? Anybody? It's going to be what? Hi, there is change in the permeability, a protein gradient, or we can say proton gradient. I'm sorry, this is going to be proton gradient. A proton gradient forms across the inner membrane, or we can very specifically say it's going to be the intermembrane spaces. So answer is going to be option D. Yes. So in this class, we have talked about the entire complete respiration in plants chapter is done. So it's a very important unit. If you're going to understand, it's going to be very easy. So today's class, we have studied about ETC, Oxford, and then we have studied about what is amphibolic pathway. We studied about respiratory quotients. And from the beginning, we have completed this entire respiration pathway from aerobic as well as anaerobic. So if you have any questions, you can just put your questions in the chat box. Yes, thank you, Roshni. You can also just comment your put your answers or questions in the comment section or in the live chat, and we'll get back to you. It's almost going to be 5 o'clock, so we'll end the session for today, and we'll be coming up with many more chapters yet again. So if you want to get the notes of this chapter which I thought for you, it will be available in the description box. Please click onto it and just download it. Thank you all of you for your time, and thank you so much for joining. We'll meet in the next classes. Thank you all of you.